Hello everyone. Today I will talk about Lab 7 RNA-seq data analysis. Here is the typical RNA-seq data processing workflow. So we start with the sequence level quality control with FastQC, and then we align those reads with HiSci 2 or other aligner. And then we'll do post-alignment quality control with Picard and RNaseq. Then finally, we will generate recounts with feature count software. The second part will do some differential expression analysis. So let's start with the small data sets. Today I will use on-demand OSC. Let's go to the cluster Pizza shell access. Okay, now I already connected. I will first go to our project directory. And here you can see the RNA-seq folder is highlighted with green color and the star with the capital RNA, not the small case, the lowercase RNA. We'll use the capital case RNA-seq folder. So next, I will go to my own directory and copy cp-r, copy the RNA-seq folder subfolder within it. Do not copy the whole folder because there are some huge data sets within the RNA-seq data folder. I don't want to waste too many storage space. Okay. Oh, I see. Because I'm in my own folder. Let's use absolute path. Okay. Now the hands-on folder is already within my own folder. And let's take a look what's inside. So there are 5, the green files are the job scripts. So we have total 5 steps. And then there is a FastQ folder. Within that folder, there are 2 small data sets. Actually, this is only one sample. And part of one sample, I just cut chromosome 1 region within the sample SRR. And this is the one ending with number 1 is the read 1. And ending with number 2 is read 2. This is the paired read of one sample. So next, let's do step 1. So that is the pre-alignment quality control. In these job scripts, we use the first several lines to require the resources from the system, like one computer, 2 cores, and the memory, time, job name, and email me when the job starts or finish, and the account we want to use. And then the first line of the job script is go to the current directory where this job script is located, and then modulo the FastQC software, and then make a directory called FastQC out. Then we call this FastQC software, tell it, please put the output into the FastQC out folder, and the input file is specified the location and the file name. So now let's run this script. S-batch. Okay, we have to go to the hands-on folder to run this script. S-batch 1. So now this script is, this job is submitted. Okay, now FastQC is done. We can tell the slurm.out file is already out, and the FastQC folder is here. So those are the HTML file. We can take a look. Let's go to the OSC on demand and go to Files. So this is FS project PAS1669. I'm going to my folder. Hands-on, FastQC out. So we can look at the output file. For read 1, everything else pass or there's some warning, but it's okay. And the sequence duplication level has a fail. But actually, this will happen almost for all the RNA-seq reads. So it's okay to make, so this sequence duplication levels is okay to fail the initial 12 base usually has the high duplication levels. And the overrepresented sequence, there is a warning, but it's okay, and the possible cells cannot find. Adapter content passed, and we can look at the read 2. Yeah, all of them passed or has some warning. So this one is okay. And we can go to next step, alignment. So we can look at the file content. In alignment. Okay, so it's basically make a BAM folder and the module load HiSci2 software and do HiSci2. This -p2 means specify that I require 2 cores to run this, 2 parallel cores to run this job. So this is because I specified 1 node and 2 cores. If I specify, for example, 28 cores, I can do the -p28 here. And I ask for the summary file. And that's the name, and that's a place where he can put the summary file in the BAM folder. And the -x means where I can look at the genome index file, the DNA index file. So the location is in the reference folder. The prefix is genome. And this is read 1, -1 is where the read 1 FASTQ file is, and -2 is where the read 2 FASTQ file is. And the last one, it means I want the output to be the same file. Okay, we can as batch. Okay, so the job is submitted. So now we can, while the job is running, we can go to the reference folder to look at the index file. It's in the RNAseq reference. So here, that is the reference file. It's called genome.1 HiSci2 index. There are total 8 files. And that is genome.fa is the genome DNA FASTA file we used. And the ending with the .fai is the FASTA index file. You have to make an index file in order to make the index for the HiSci2 aligner. Now let's look at the alignment results. So the BAM file is already out. We can first look at the slurm out file. Let's list it according to the time sequence, and this slurm file is the one I just submitted. Okay, so here is the HiSci2 summary and statistic. Totally, in the FASTQ file, we have 2 million reads and aligned concordantly or disconcordantly. Let's look at this file. It's easier to look. There are 8% has no alignment, and 80% of the read. The blue one means both read 1 and read 2 uniquely aligned to only one location, and the red one means both read 1 and read 2 aligned can be aligned onto multiple region on the genome. So we also call it multiple mappable reads. And there are about 0.8% of the reads aligned disconcordantly one time. So among those disconcordantly reads, you just double this number. So double 18,836, you get, you double this number, 189,220 reads out of those unaligned reads. If we make them unpaired reads, then out of those unpaired reads, 37% not aligned, and 35% aligned uniquely once, and 26% of them aligned multiple times, can be aligned to different regions in the genome. So the overall alignment rate is 96%. It's pretty good. Next, let's go to step 3 to process the SAM file converted to BAM file. Let's look at the content in the step 3. The header are all the same. So in this step, we modulo the SAM2 file, and we converted from SAM file to BAM file, and then sorted the BAM file into the sorted BAM file, and then index this sorted BAM file. That's S batch 3. Okay, so this job is submitted. While it's running, we can take a look at the output BAM file header. So this is the BAM file header. It tells you the version of the tool, and also the sort order is by the coordinate. It's longer than this, so I just skip the middle part. And the last several line can tell you which program has already run. So this has run the SAM2s, has run HiSci2 and the SAM2. So next step, the step 4 is the post-alignment quality control. In this job script file, we make a RNAseq folder and tell it the rainbow bad file location and the bad file location. This is just to tell RNAseq I'm interested in a list of genes. Those are the ribosomal gene, and I want to look at the contamination level of those genes. How many reads are aligned to ribosomal gene and how many are aligned to what we are really interested in, the messenger RNA. And then in order to run the RNAseq, this is a Python package. So we have to run the Python. I usually use the Python 2 version. And you need to export some Python paths to your working environment in order to use this tool. So I just used the 3 commands within the RNAseq. The first one is junction annotation. It will tell you how much junction reads are there, and out of them, how many are novel junctions, how many are known junctions. And the second one is read distribution. The third one is split the BAM. And this is tell you the ribosomal RNA contamination. So in the output file, okay, let's run this first. Let me see. Yeah, the previous one is already done. Let's look at the BAM file in the BAM folder. Yeah, the BAM file and the sorted BAM and the BAM index are all done. Let me look at the output file. Okay, and let's submit the job 4. Okay. So for job 4, in the output files, once the job is done, you can tell it will give you a summary. What is the total number of splicing event and the known splicing event or partial novel splicing events. I think I have some example. Yeah, here you can see it will create some PDF file plot, tell you out of all those splicing junction reads, the known one is about 70%. Complete novel means both of the exon are unknown. Those has 18%. And the partial novel means only one exon is new. So those are 12%. And the read distribution, this one will tell you how many reads are distributed in the CDS region, how many in 5' UTR or 3' UTR, or intron or upstream 1 kb region. Yeah, this is pretty normal. Most of the reads are distributed in the CDS exon region. And also the ribosomal contamination. So it will give you different BAM files. In the first one are the reads that mapped into the gene list. Here is our ribosomal bed file. There are only 2,000 reads consumed by ribosomal, so the ribosomal removal is pretty clean, pretty completed. And the not ribosomal reads are the majority. And there are some reads failed the QC check, is unmapped reads. Okay, let's look at our job is probably already done. Yes. So we can look at the slurm out file, the latest one. The first part is tell you about the splicing event and the splicing junctions. Then this part is tell you about the ribosomal contamination. And in the RNAseq folder, you will have those files, the BAM file, the read distribution, and those junction plots. Now let's go to step 5. This is the last step. So in this file, in the job we just submitted, it make a new folder called feature counts, and then it used the software I already installed in our classroom folder, subread software in the Linux distribution, and use the feature counts command within the subread package. And the first one the -A tells the software where it should look for the GTF file. And then it tells the software I just want to count the primary aligned reads, and tell it where the output file should be located in the feature counts folder, and where the BAM file, the sorted BAM file, the input file should look for. And that is what a typical output file look like. It give you a summary of the feature count setting and the running summary. So it's very important to use the correct annotation file. Here we use the human grch30a dot version 96 GTF. If we look at this file, the GTF format, it's an annotation file, defines genes, transcripts, and exon. Here is the header, and for example, the right one here is corresponding to this gene, this transcript, ENST 456328, 45632A. It's defined where each exon start and end. And this one here is corresponding to this transcript, ENST 0000450305 is another transcript. So this GTF file lists all the gene and transcript annotation, the start and ending point of each exon. So what feature count is doing is based on the BAM file, the aligned read file, and then it will compare it with the GTF file. With defined gene A and gene B region, it will determine whether those reads are aligned belong to gene A or gene B. Sometimes it's easy to count, like the first situation, but sometimes it's ambiguous. This read can belong to both gene A and gene B because these two genes overlaps with each other. And there are also some situations that one read can map to multiple locations. Those are the multiple mappable reads or multiple aligned reads. This happens already in the HiSci2 process. So usually what we do is disregard those reads. But some software like RSAM, they also use EM algorithm to calculate the most possible alignment for those multiple mappable reads. And also some reads aligned to the intron region, so that's no feature. So here is the feature count results summary. So there are majority of the reads are assigned, and there are some reads are unassigned, and they have different reasons not to assign those reads. Some are due to ambiguity, like this, the ambiguity. Some are due to multiple mapping. We don't have multiple mapping issue because in the command feature counts, I ask it to just use primary alignment. So there is no multiple mapping reads. And some are no features. They are maybe intron or intergenetic region. Some are unmapped. And if we look at the feature count results, then it will give you the each gene ID. I asked it to align at the gene level, but not the transcript level. So it will just count at the gene level. And that is the gene, the chromosome, and the exon start point and ending point, and whether it's a positive strand or negative strand, and this gene length, and how many reads align to this gene. The red color is the number of reads aligned to this gene. Okay, I think our job should be done. Yes, let's first look at the Slurm output file. Here is the process of running the subread. It will record the feature count setting. So this is input only one BAM file, and the output is called this feature ending with the feature counts. And the summary is summarized in the one ending with dot summary. Annotation file used, version 96 GTF file. And the thread used only one thread. And the multiple alignment reads are not counted. Only count the primary alignment only. And that is the running statistic. How many alignments processed. Runtime successfully assigned alignment, 53%. And now let's go to the feature counts folder to look at the summary file. That is the number of assigned reads and those unassigned reads. And let's look at the result file. Yeah, this is the same as what I just showed you in the PPT. Okay, so this is small example, small set already finished. And our homework is also based on this small set. But in the real life, we are not usually just do one sample. We usually do multiple samples. So here, in the full set folder under the RNAseq folder, there are total 14 samples and 28 FASTQ file because each sample has two reads, read 1 file and read 2 file. So those are 7 pairs prostate cancer samples, pre and post treatment, drug treatment. And if you want to run the pipeline, the full pipeline, this is very similar to the real production pipeline that we run in a bioinformatics core. Then you can make a directory in the Scratch folder. Please pay attention here. It's not a project folder. Our project folder doesn't have such a large space. So you have to create your own folder under the Scratch folder, under the PAS 1669 project folder, but within the Scratch space, and create your own folder. Then you can go to that folder to copy these full sets pre-run into the folder, and then you can go to the pre-run folder and just type dot slash config and run human.sh this shell script. Then it will run the whole pipeline. I also list the result in this post-run folder under our project classroom directory. If you want to check the result, you can check it there. So next, I will just go through from here what is the pipeline look like. This slide just explain why we want to create a Scratch directory, because we have limited file space. And also there are some file deletion policy in this Scratch directory. It only saves the file for 120 days. So this workflow is like this. We have a file with list of the sample FASTQ file. And also we have our genome reference and all the accessory files we needed in the configuration. So we have a configuration file to list what are the input sample, what are the genome reference, and what are those ribosomal bed file and extra. And then according this list of sample and the same PBS script or Slurm job scripts with the same workflow will run per sample. So each sample in this file will run the same job scripts. So this one we can make sure that each sample are processed identical. And then we have a script to gather the results and to do some differential expression analysis with our code. So within the workflow, it mainly has to contain the same part I just showed you in the small data sets. The FastQC, HiSci2, RNAseq and the feature counts. We also have the convert BAM step, just we didn't list it here. So the configuration file has listed all the file paths, name, reference, gene annotation, GTF file, extra. And the sample file has all the sample name. It's the same as the one I showed in the command line interface lab. It has 3 columns and is tab delimited. First column is read 1, and the second column is read 2, and the third column is the sample name. And the PBS script is the commands to run or the workflow to run on each sample. So here is a sample file. This is tab delimited. The first column is read 1, second column is read 2, and the third column is the sample name. This is the configuration and the run.sh file. It will tell you what is the project directory. It should be your current working directory. And which PBS script. I still use the old PBS script. Nowadays, the OSC will automatically convert the PBS script to Slurm script. And the sample list, the reference genome for HiSci2, the GTF file for feature count, the ribosomal bed file and the bed file for the RNAseq ribosomal contamination level control. And finally, this will submit the job to run the submit PBS.sh file. So this is the submit PBS.sh file, and it will make some new folders. And while read the input file $1 here, the input file is samples now. it will read the three columns. The first column is the FASTQ1, and the second column is the FASTQ2. Third column is the sample name, and it will echo the sample name, and the Q sub is the same as the S batch, and it will tell the configuration file the R1 is equal to this variable, R2 is equal to this variable, FASTQ2 variable, and also the output file and the reference file, the ribosomal bed file, where to look at those reference files. And the -n is the job name, and the -o is output the log file. So finally, the last variable is the PBS script name. All those are already identified in the configuration file. So next is the PBS file. We call it the HiSci2 RNA-seq PBS file. It will specify the node to use, one node and 28 cores, and what time for one sample. It do not need 50 hours, but we just let it to require enough time. And then that is the 5 parts, same as I showed in the small hands-on dataset. The FastQC quality control, and HiSci2 alignment, BAM file conversion, post alignment quality control by Picard and RNAseq, and finally feature counts to generate recounts. So after all jobs are successfully finished, you can gather the result by Q sub this generating the final report script.